let's test this and see if it drops out. No, I think you can all hear me. And as she said, if you all want to know about sailing Viking boats, then you can also ask me afterwards. Is it dropping a little bit? Because then we'll let you know. All right, I would like to tell you a story. And this story, as every fairy tale, starts way, way back, once upon a time, in a very different kind of Earth, when it was atmosphere full of CO2, very humid, very barren, a bit like in here today. And there was no life. There was a little bit rustling in the oceans at that time, but not any life as we know it, with plants and mammals. And then something wonderful happened in the oceans, and that was that one singular cell found out, or evolved into, using photosynthesis to produce energy for itself. And that meant that it changed the atmosphere from being predominantly CO2 to being full of oxygen. And that's what we need as humans and all other mammals. So that was really great for us, that we could finally get to rise into power 3.7 billion years later. So how does this lake full of algae, that's by the way from my hometown in Denmark, and this lake always looks so inviting, but it's always full of algae, and you're not allowed to swim in it because you're gonna breathe in all, or like swallow all the algae, and that's not good for you. How does these algae and what they did for us back 3.7 billion years ago, how does that relate to a barren landscape like this? So this is from The Martian. We haven't yet landed there, as I'm sure most of you are aware. This is Matt Damon. And he's very nice, also from behind. So how does this lake match? How does this lake match with this? Well, we can use algae to make a life support system. And this was some of the things that Jennifer talked about before. We have to keep our humans alive in space, and it's not very easy. But I'm not going to go into all of the microgravity radiation part, but only the very bare minimum, and that is that we actually need to eat and breathe. So back to bare necessities. So a life support system is a system that provides everything that you need, and that's air, food, water, waste, and safety. And I write safety in parentheses because I know nothing of that. So if we just exclude everything except for air and food, and then maybe you already kind of grasp the connection to the algae in the beginning of the story. Because algae, they can substitute what we currently do. So these are pictures from the International Space Station, where they have to bring up everything that they need. So they have to, well, right now at the International Space Station, they're also pretty good at recycling, but they still have to bring up every food item that they want to eat. They have to package it very clever, but still it takes a lot of mass and a lot of money to bring all of this food to space. And so maybe we could use what originally kickstarted life on Earth to kickstart life on other planets. I wrote my my master thesis on life support systems, but basically it's all about how to keep people alive in space. It had a very fancy and long title, but it boils down to that. So if we want to go to Mars, and Mars was the topic of my thesis, we have to bring a lot of stuff, as I just showed you in the previous picture. So the current estimate is that if you want to bring everything you need to Mars, you would have to bring 8,000 kilos per year per person. And that sounds like a number. I don't know if it makes any sense to you, but it sounds like a lot. It gets worse when you look at the prices per kilo. This estimate is from SpaceX, and they are the absolute cheapest estimate, and they haven't done it yet. They're just saying they can. So this is the very minimum of both things, the bare minimum of what you need and the minimum of what it costs. And then the minimum duration would be 545 days, and that is if you just go there and like tap on Mars and then go back. But if we go to Mars, we probably want to stay longer. So this is, again, very, very minimum. It would probably be several years. So back to the lake with the algae that ruins my summer holidays and makes me unable to swim in my lake. We could take these algae and we can compress them down to a very tiny kind of thing. I have brought them with me, so if anyone wants to taste afterwards, because they are edible, they look like this. There's a little tiny pill. You can probably not see it in the back. You can come and look at them afterwards. then we can use them for two purposes, which are coincidentally also the two purposes that we used them for in the very beginning of life on Earth, which is supplying us with oxygen and eat them. Because, okay, maybe humans haven't before. Well, humans haven't eaten them before, but other mammals have, especially fish. It also tastes a bit like fish food, so it makes sense. This is what I did in my master's thesis, because someone found out to compress this lake into a very high-tech looking fancy light system here. This is called a bioreactor, and it's a controlled lake. So you have water, you have CO2 flowing in, and then you have the algae inside, and then O2 comes out, and you can breathe it, and then you can take algae out occasionally and eat it if you're up for that. And so the idea is that astronauts would have to have some of their food supplied by algae. We can talk about if it's a good idea or not afterwards. So this algae in particular is called Chlorella vulgaris, it's the Latin name, and it's just one of several algae or cyanobacteria that does the same purpose, that takes CO2 and makes food and oxygen. So it's edible if you treat it, and it's small in size, which is its advantage over other plants. Like, you might also know that NASA and ESA are researching how to grow salad in space, which is also necessary and really cool, but salad take a long time to grow and it's pretty big. So if we could grow something faster and eat it faster, then it would be better for us. And then they're really hard to kill, so that's also why they're in my lake. You can't get rid of them, and that's beneficial in space, so you don't kill them with radiation and whatnot. And then it's also a commercial product, which is the reason why I have this little pill glass, because you can buy them in a pharmacy, because they're good for your digestion. It's a hard word to say. And so there are multiple partners that are interested in this, not only space. The only problem for using it in space and eating it is that it's so full on protein that you cannot eat more than 30% of your daily intake of food from this. I'm sure you wouldn't want to anyways, but you definitely can't, because then you get sick. So these are the constraints of Chlorella vulgaris. So what I looked into, investigated in my thesis was how to make the algae grow really fast so that it would produce enough oxygen for 6 astronauts, for example, or 4 astronauts, and enough food to have 4 astronauts eat 30% of their daily intake per day. And that sounds like a lot. It turned out to also be a lot. And the things that influence how fast this algae grows are the design of that fancy system I showed you before. What environment you give it, like how much light do you give it, how much CO2 do you supply, the nutrients, the temperature, and how much and how often you take out algae. And all of these things I put into a computer and simulated stuff. It was pretty complicated for me at least, but out of all of that came a really depressing number. And to the left is the weight, and it's a special kind of weight. It takes into account also how much energy a system uses. It's called equivalent system mass, if people are aware of what it is. It's basically what you measure how good a system is to bring into space. How much it weighs is how much it costs. So the current life support system design weighs 7,375 kilos. And after bringing all of the numbers I had from all of the other factors I showed you before and designing a photobioreactor system where they would get oxygen and food from algae, it weighed 16,419 kilos. And that's really, really bad. And the reason it did that was because the photobioreactor was giant and it required so much water. And I think if people have written a thesis before, this is usually what comes out of it. You spend half a year and then in the end you're like, didn't work. But of course you can't end your thesis like that. So of course you have to be like, what could we do to make it work? And this is how we enter into the second half of the presentation. What could we do to make it work? So as you might see here underneath it says growth rate. These algae, they grew like 0.4 grams per liter per day. And that's why we needed so many liters, that it wasn't beneficial. But what can you do? Think about it. You could increase the growth rate. So as soon as you go to 2 grams per liter per day, which has actually been done in other labs, just not the one I was in at the current moment, then you're kind of close. And if you could go even higher, if you could go to 4 grams per liters per day, then it might actually, at least hypothetically, be possible and beneficial to use a biological component. So, I'm not saying algae is the holy grail of space exploration, especially if you dare come up and taste it afterwards, which I all encourage you to. I mean, astronauts have to be really strong and cool and do whatever you tell them, but I think there's a limit, and that might be it. But in any case, the point holds. To use biology in space is a valid and a good idea to pursue, because as soon as we manage to know how to grow and kind of reinvent agriculture in space, then we can also use it back on Earth. As Jennifer also told you, everything on Mars has a benefit on Earth. And if we can combine the best systems, so maybe we can use algae for oxygen and salad for eating, or something like this, hybrid systems that uses technology like electrolysis and chemistry and physics, but also biology, then we might be one step closer to actual space exploration. So, this brings me, as I said, to the second part. Ooh, I hope I have time. This is about the prospect of designing plants or organisms, which is another project I did. It was not my thesis. It was called Space Moss from Copenhagen University, and it was the Kickstarter of a very long sequence of projects made by students at my university that was about how to do interdisciplinary projects between students from business, from physics, from biology about space exploration. And in Space Moss, we wanted to make a proof of concept, and that was basically because we wanted to tell our professors that they were wrong, that we could do stuff without money. So we were self-funded, we were I don't even know, outreachy. That's why I'm here today. I did a lot of outreach on this project. And we wanted to shove it in their face that students could do at least a proof of concept, that we could show that it was possible. And what we chose was to genetically modify a plant to produce something that we could use and also be better suited for something that is in space. We chose temperature because it's very cold on Mars. So what we did was we looked out into a forest. Well, it sounds very exotic. We looked in a very long database on the internet for genes, but this gene that we found is spruce buttworm, which is a pest in the North American forest. It eats the pine trees. But as algae is a pest in my lake, this little bug was also a miracle to us, because it can also eat the pine trees in the winter. And it can continue eating the pine trees even though it's minus 30 degrees Celsius. And that's because it has a protein inside it that inhibits the crystals from growing and puncturing the cell walls. And that protein we really wanted, so we ordered it online. That's what you do, apparently. And it came in a little package. And then we also ordered a little jellyfish gene that's called, I don't know, I hope there's no biologist, because I'm a physicist, so I actually don't know anything about this. No, but we did this project with the biology student. So they were behind the whole thing. I pipetted mostly. But what we did was we took a fluorescent jellyfish and we stole one of its genes, and it works in the way so that if you have successfully put this protein into, we use moss as our plant, if you successfully put that into moss and you did it in a special way, you can also see fluorescence light shining from your cell if you've been successful. I don't have time to go into why, but I have extra slides if anyone wants to talk about it afterwards. So when we looked at our moss cell, can you barely see it? It's shining. I don't know if you can see it, but it is. And that means that we successfully put the gene, the antifreeze protein, into the moss cell. That doesn't mean that it works, so we also had to freeze the moss down. And it's a really bad picture. Can't see it, actually. Anyways, so ignore the picture, we go back to this. What I can I can just tell you that what we saw was that after 24 hours in minus 20 degrees, the moss that we had successfully put the gene in was less dead. It sounds very vague, but it was not like a green flowery moss, but we also checked with autofluorescence, which is the light that plants emit, and it had certain patches that were still alive, whereas our non-genetic moss was completely dead. So we did that, and we went to Boston and presented it, and everyone was really happy, and our professors made it much better effort to next year tell everyone that moss grows really slowly and our project was a one-off and that we should do better now. And that's fine, because that was what we wanted. We wanted to start like a student movement at our school. So a new team the year after, and the year after, and the year after, so we're still going. I'm not in it because I'm here, but everyone else is still working on this project. And they did something much smarter, which was they used bacteria and the cyanobacteria to make a co-culture. I'm gonna be finished very quickly. This is the last slide. Which is a mix of two organisms that usually wouldn't live together, and then you separate them by kind of a tea bag so that they can't mingle, but they can exchange water and nutrients and all of this. And so the cyanobacteria is like the algae I showed you before. It uses the CO2 and light to produce sugar and oxygen. And then they had, this is very vague. Then they genetically modified the other bacteria, the Bacillus subtilus, to produce plastic, bioplastic. And the idea with a co-culture is then that the cyanobacteria provides the sugar and oxygen that the GMO bacteria then uses and then produces plastic. And the idea with that is that you can make your own plastic and potentially 3D print the things you need in space. And thus, in the end, we showed over 2 years at least that we could modify plants or organisms to survive cold temperatures, to produce something beneficial like plastic. And at the same time, I also got the opportunity to travel all over the world and talk about it, which I really like. So I think I'm out of time. I really encourage you all to come up and taste algae. It tastes you get used to it after a while, so I guess. Or you can take it as an astronaut test. So if you're strong enough for this, you're strong enough for space. And if you want, there's also on the mirror down in the back, there's a cartoon that we produced hanging on the mirror. That explains everything in like a cartoony way about the Space Moss project. So I hope you all liked it. Is questions?